Hello students, welcome to Toppscholars, the smart learning app. In today's class, we are going to solve a few questions which will help into your NEET examination. So let us begin. So students, let us begin with the paper analysis for biology and we understand the difficulty level here. Okay. So the level of questions which you would find into the section A, the difficult questions will be around 11.4% here, right? And the medium will be for 42.9%. And you have around 45.7% of questions which will be very easy for this paper for this section A, okay. And for your section B, into section B you would find you have a medium level of questions that would be asked which will be around 46.7%. For difficulty and easy, they have given a very equal ratio that will be around 26.7% here, okay. And next over here difficulty level for your section A and B, that is your class wise distribution of your section A and B. So you will be getting around class 11, from that you will getting around 46% of your questions, and from class 12 you will be getting around 54% of your questions. And if you go for the difficulty or the levels of questions for 7 May 2023, it would be, it was around easy questions, they were around 54%, medium questions they were around 34%, and difficulty level that is around 12% here. So students, first let us go through the syllabus once. We have a few topics that is from your class 11 which are very important, and here we have the list of questions or number of questions which will be asked from the topic, right. So first we have diversity in living organisms, right. Into this we will be talking about plant kingdom, animal kingdom. Each of the topics will have 3 questions, right. Then we have structural organization of animals and plants. Again a very important topic because you will be learning about morphology of flowering plants, anatomy, about the cockroach, and the structural organization in animals, that is about your animal tissues, different types of tissues. For this we have different number of questions. Except for cockroach, each of the following topics will have 3 questions, okay. The next important chapter that you have is cell structure and function. Into this topic we are learning about each and every detail of the cell, right. So we have cell cycle and cell division. We will be learning about the biomolecules. Each of the topics will have 3 questions each. Then you have the cell, which is the unit of life, right. Into this topic you will be having 2 questions each. Then we will be learning about the transport in plants and about the mineral nutrition as well. Transport in plant will have 3 questions, mineral nutrition will have 2 questions. Then about the photosynthesis and respiration, from photosynthesis 3 questions and from respiration 2. Last one that we have here is plant growth and development, that is 2 questions each. So the next chapter that we have here from your last 11 is human physiology, right. So human physiology, we will be learning about the whole human body. The various topics that we are learning here is digestion and absorption, that is majorly your digestive system. You will be asked about 2 questions from digestive system. From breathing and exchange of gases, there is only 1 question. From the body fluids and circulatory system, we have 2 questions. And for locomotion and movement, in that we talk about muscles, skeletal muscles, how the movement is happening across the human body. From that we have 2 questions. Another important topic that we have neural control and coordination, that is majorly about our nervous system, synapses, the action potential, the reflex actions, right, and about our sensory organs. So we have 2 questions from them. Then we have chemical coordination and integration, that is majorly our endocrine system, how the hormone system are working to find communicate across your body. From that we have 2 questions. And last that we have your excretory products, from that again 2 questions. The next topic is from your class 12 actually, that is your reproduction. So into reproduction we have sexual reproduction that we find into the flowering plants, that is a separate point which we will be having 2 questions from that. Human reproduction and human reproductive health, both of these topics will carry 3 questions or 3 questions each. Again a very interesting topic that we have is genetics and evolution. Into this chapter we have principle of inheritance and variation. A very important topic, we learn about Mendel and how the experiments were done by Mendel. From that you have total 6 questions, okay. And another interesting one, that is your molecular basis of inheritance. From that we have around 9 questions. So you know how important this chapter is and how in detail you have to learn about this here. Then you have origin and evolution, only 1 question from this, okay. Next that we have is biology and human welfare. So in this we are going to learn about the strategies for enhancement of food production, that is plant breeding, how different types of plant which are bred depending upon the characteristic features which we require in them. From that we have just one question. Human health and diseases, 3 questions. We learn about lot of diseases, communicable disease, non-communicable, and a variety of pathogens here. And strategies for enhancement in food production, here we'll talk about animal breeding. So again, one question from this one. Then you have ecology and environment, which is coming under the category of biotechnology and its application. Biotechnology principle and processes are very important chapter. We learn about lot of biotechnological techniques, recombinant DNA, and various ways into which biotechnology can help into medicine, okay, and also about its application. So you have 2 questions from that. Organisms and population, you will be getting 4 questions. Ecosystem, biodiversity and conservation. So for ecosystem you have 2, biodiversity conservation you have 3, and environmental issues you will be getting 4 questions. A very simple topic and you can get 4 questions from that which you can easily answer. So students, let us begin with the question here. So the very first question which is given to you, you are provided with 2 statements. The first statement is, during the G0 phase or G0 phase or the gap 0 phase of the cell cycle is metabolically inactive, right? The second statement that we have is, the centrosome over here undergoes duplication during the S phase of your interphase. And the third statement that you have is, in the light of the above statements, you have to choose the most appropriate answer from the given options below. So the first option that we have is option A, both 1 and 2 statements are correct. Option B, statement 1 is correct but 2 is incorrect. Option C, statement 1 is incorrect but statement 2 is correct. And option D, both statement 1 and 2 are correct. So here students, we are talking about the G0 phase. So G0 phase is not a metabolically inactive state, right? Why? Because here you would see this is our G0 phase, right? So into the M phase, we know that we have our mitosis that is going on, right? And from the M phase, the next phase that we have is G1 phase and we have the G0 phase. So into this G0 phase, our cell is going to prepare a lot for the S phase, that is the next phase, synthesis phase. So as there is preparation going on, so it will be metabolically active. Correct? So that is the incorrect statement. So G0 state is not a metabolically inactive state. And centrosome, yes, they can duplicate into your S phase. So we have a similar question in Toppscholars NEET mock test. We have to identify the incorrect statement about the G0 phase. Undergo the differentiation to perform a specific function. These cells are metabolically inactive, okay, and they divide only after getting a signal to divide. All of these cells. And then cell differentiation in the quiescent stage. Over here, we are talking about cell differentiation. It is a process where the cell is going to determine what is the specific function or specific feature of that particular cell. So it is comprised of the differentiation phases. It grows in size and assume a particular shape depending upon a particular function that is going to be performed. So here the correct answer would be all of these. So into the above example, these cells are metabolically inactive, right? We got that into a mock test question, a similar question. And here all of these questions are correct where you say that the cell differentiation in the quiescent stage comprises of differentiation phase, grows in size and assume particular shape and have a particular function. So here students, the correct answer will be your option C, where one statement is incorrect but statement 2 is correct. So the next question here is about the process of appearance of recombination nodules occurs at which substage of prophase 1 into meiosis. So the option here is pachytene, option B is diplotin, then we have dikinesis and zygotine. So now recombination is a process which is a very common method which you would find into a meiotic kind of a cell division. Why? Because here you would observe the genetic material which getting exchanged. Genetic material is exchanged into this particular stage, right? That is the reason into meiosis you are getting a lot of variation, a lot of variety among the cells. So recombination nodules which is observed, it observes into the pachytene stage of your prophase 1. Meiosis again is divided into two divisions. So we have meiosis 1 and meiosis 2. Meiosis 2 is a lot similar to mitosis, right? It is exactly the replication of mitosis, but into meiosis 1 phase, there are different steps or different stages which are not repeated into the meiosis 2. So here you have into meiosis 1, recombination. So where exactly the recombination would occur? It would happen into the prophase. Now our prophase again is divided into few stages. We have leptotene, zygotene, pachytene, diplotene and dikinesis. These are the 5 stages in which your prophase 1 is divided. Out of these phases, the question asks where you would find the recombination nodules which are observed. So here into this stage, into the prophase 1, you would observe into the pachytene phase. Because into the pachytene, here we can see that the genetic material which is getting exchanged, that is the step of recombination. Into the leptotene, they are just arranged, the sister chromatids are arranged, right? Into the zygotene, they are forming together with the homologous chromosome. But into the pachytene phase, we would see the recombination that is beginning. Into the diplotene, the recombination is completed, and in the dikinesis, they are separated. So here the answer would be your pachytene. Into the pachytene, the step where the recombination occurs, we call it as the chiasma. So the correct answer here is option A, pachytene. So the question here is, unequivocal proof that DNA is a genetic material was first proposed by which of these scientists? Option A is Alfred Hershey and Martha Chase. Option B is Avery, MacLeod and McCarthy. Option C is Wilkins and Franklin. And option D that we have is Frederick Griffith. So here this experiment that the DNA is actually our genetic material, that was first proposed to us by the scientists called as Alfred Hershey and Martha Chase. So it was a very famous experiment which was given to us by Alfred Hershey and Martha Chase, commonly known as Hershey's-Chase experiment. So what they did into this experiment was, they took a bacteriophage. So what is a bacteriophage? So bacteriophage is a virus that is going to infect a bacteria. Okay, so it's a virus that infects the bacteria. But here into this bacteriophage, they had added some radioactive elements. So they use S35, right? S35, a radioactive element into the bacteriophage. And they infected the bacteria with that bacteriophage. So into the first slide of the image, you can see that the bacteriophage, one is labeled with radioactive S35. The bacteriophage is infected along with the bacteria. And the other radioactive element that we used was P32. Okay. Now here, once the bacteriophage has infected the bacteria, they allowed the binding of the genetic material which is present into the bacteriophage to bind with the genetic material of the bacteria. Okay. Once the blending was done, they allowed the bacteria to grow and increase in number. After that, they undergone the process of centrifugation. So into centrifugation, it was observed that into the first A, like let's say if we consider this as a left side image as the A sample. So into the A sample, there was no radioactive S35 detected into the cells. Why? Because sulfur is not present into the genetic material. So hence, it was not embedded into the bacteriophage genetic material, neither it got into the genetic material of the bacterial cell. So hence, S35 was detected into the supernatant and not into the pellet. Because into the pellet of the centrifugation product, you will get DNA. Whereas, on the other side, into the other side of the experiment, when the bacteriophage was blending, right, here the genetic material contained P32. Phosphorus is present into the DNA of any genetic material as into a sugar phosphate backbone. So that was observed into the sample. So P32 was detected into the cell. So radioactivity was present into the pellet and not into the supernatant. So this confirmed that your DNA is acting as a genetic material, and this was proposed to us by Alfred and Hershey into the experiment known as Hershey's-Chase experiment. So here the correct answer would be option A, that is Alfred Hershey and Martha Chase. So the students, the question here is, large, colorful, fragrant flowers with nectar are seen in option A, bird pollinating plants, option B, bat pollinated plants, option C, wind pollinated plants, or option D, insect pollinated plants. So now all of the features that we are observing, the flower being large, colorful, fragrant, these along with the nectar, these are seen into insect pollinated plants. Why? Because here the insect will be attracted to all of these attractions, like large colorful flowers, fragrant flowers is going to attract the insects so that they can be attracted towards the nectar that they have. And when they are attracted towards a flower and they are trying to absorb the nectar from the flower, from that also the pollen grains which are present onto the flower will fall onto the body of the insect. And when that insect will be traveling for the nectar for any other flower, these pollen grains which were present onto the body of insect will be transferred to the female reproductive organ, that is the stigma of the flower, that is for the another flower. So that is how insect pollinated plants would be transferring the pollen grains. So here the correct answer that we have is option D. The rest of the options are also incorrect because birds, when they are pollinated by birds, you do not require large colorful fragrant flowers with nectar. Even that's same for bat. And for wind pollination, ideally we require a very different feature. Those pollen grains should be very light. For wind, you don't have to use such attractants, right? So the correct answer would be option D. So here the question is, what is the role of RNA polymerase 3 in the process of transcription in eukaryotic cells? The option A is transcription of tRNA, 5S rRNA and snRNA. Option B is transcription of precursor of mRNA. Option C is transcription of only snRNAs. And option D is transcription of rRNAs 28S, 12S and 5.8S. So RNA polymerase is ideally of different types, right? So you have 1, 2 and 3. And these are majorly the major products, that is your rRNA, mRNA and tRNA. So depending upon the polymerase that is being used, you would get different product. Because this is actually the process of a transcription where your DNA is used to produce different types of RNAs, right? So when you are going for RNA polymerase 1, it will be used to produce rRNA, right? And that will be done into the nucleolus of the cell. Whereas the RNA polymerase 2 will be produced to give us mRNA, messenger RNA or snRNA into the nucleoplasm of the cell. Whereas the RNA 3 or RNA polymerase type 3, it is used in the process of transcription which helps to produce tRNA, 5S RNA, that is your ribosomal rRNA, U6 and snRNA, which also happens into the nucleoplasm of the cell. So here in the question we have been asked about what is the role of RNA polymerase 3 into the process of transcription. So all of these RNAs will be produced with the help of RNA polymerase 3 into the nucleoplasm of the cell. So here the correct answer would be your option A, that is transcription of tRNA, 5S rRNA and snRNA. So students, here we are provided with two different statements. Statement A is, or statement 1 is, the forces generated by transcription can lift a xylem-sized column of water over 130 meters height. Statement 2 is, transpiration cools leaf surfaces sometimes 10 to 15 degrees by evaporating the cooling. Now, in the light of the above statements, we have to choose the most appropriate answer from the options given. Option A over here is both statement 1 and 2 are correct. B is 1 is correct but 2 is incorrect. C is 1 is incorrect but 2 is correct. And D over here is both statement 1 and 2 are correct. So the forces actually which are generated by your transpiration, right? Into the transpiration process, what's happening? As the temperature is increasing or for the process of photosynthesis, the stomata cell is going to open, because of which the water will be lost by the plant in the process of transpiration, right? Now, when the water is lost by the plant, the forces are going to be generating the water from the xylem column, right? So these forces which are generated because of the transpiration of the water loss, they can lift the xylem-sized column of the water over 130 meters height as well. Because of the transpirational forces, as you can see, the water will be moving upwards. Ideally, this is just the game of osmosis which is happening here. So let's say if the plant cell is losing water or the leaf is losing water because of evaporation. So the concentration of oxygen reduces into the leaf region, right, because the water has evaporated. But the concentration of the xylem level is still high, or the water level in the xylem is still high into the stem. So the water will be diffusing from the higher concentration towards its lower concentration. That means from the stem to the leaf. Now here the concentration is reduced for the stem because the stem has provided the water to the leaf, but it is still high into the root cell. So again a concentration gradient is created and the water will be travelling from the roots to the stem. The same gradient is created when the root levels or the water in the roots reduces compared to the surrounding soil. So the water will be absorbed from the soil by this osmosis gradient or osmotic gradient. And this is the reason you would find that your xylem-sized column of water over 130 meters of height as well can also generate that force and water can be transported. The second statement says transpiration is going to cool the leaf surfaces sometimes 10 to 15 degrees Celsius by evaporating cooling. Yes, because as the temperature reduces, the water gets evaporated. That will help to reduce the temperature, just like our body sweats. So here the correct answer is option D, because both 1 and 2 statements are correct. So students, here into this question, you have to match list 1 with list 2. So into list 1, you are provided with few interactions, and into the list 2, we are provided with the species. Okay. So here we have mutualism, commensalism, amensalism and parasitism, and we have a list of species which are given here. So depending upon that, we have to choose the correct options, going for a correct combination over here. So we have mutualism. Into mutualism, this particular sign is going to indicate that both the organisms are benefited out of it, right? It's a kind of a mutual relation or mutual correspondence between two organisms. So this plus sign indicates both the organisms would be benefited. Into commensalism, you can see that one organism is benefited and other organism is not having any effect over that. So you can call it as an unaffected, right? So again, there is no loss into this situation because only one is benefited, but the second one is not going through any effect because of that. Amensalism, you would see one is harmed while other organism remains unaffected. Whereas into the last one, that is a parasitism, only one is benefited and the other organism is completely harmed out of it. So for example, ticks and fleas survive onto the body of a dog. So that is a relationship or a parasitic relationship because the ticks are benefited from the blood they are getting from the dog, and in return the dog is harmed. So here we have the correct combination for option A. Again here we have to match the list 1 with list 2 with respect to human eye. So here we have the list, that is first one is your fovea. So fovea is actually that region of your eye which is going to give you the greatest visual, or you can say just like a microscope is going to resolve an image for your vision, but for microorganisms, fovea is going to show this do the similar role, but for our eye, so that our visual acuity, that means you will be watching with a proper accuracy and it will be a resolved image. Okay, so A will be paired with 3. Okay, with this actually we can learn how to eliminate the options as well. As you can see over here, just by knowing the first option, you can eliminate the rest of the 3 because you don't have any other option showing you this combination, right? Then second that we have is iris. Iris is actually the visible colored portion that we see in our eye, which gives a color to your eye, brown eye or black eye. And that is going to actually regulate the diameter of your pupil depending upon whatever light is coming into the eye, right? Then we have a blind spot. So it is a point into your optic nerve which is going to leave the eyeball, and the photoreceptor cells are absent over there. Photoreceptors, that means these cells are going to recognize the light. Our eye actually functions just because of the principle of light, right? So here, as you don't have any photoreceptor cells, they are absent. So if any light comes even onto the blind spot, the image is not going to travel up to your brain. Then we have sclera. Sclera is actually the external layer of eye which is formed because of your dense connective tissue which holds the different types of muscles which are present into your eye. So here, as we have already eliminated rest of the options, but still we know that our correct answer is option D. So into this question, which of the following are not considered as the part of the endomembrane system? The first option is mitochondria, option B is endoplasmic reticulum, option C is chloroplast, and option D is Golgi complex. And the option E that we have is peroxisomes. So here we have to choose the most appropriate answer from the options which is given below, that is A, C and E, B that is have is A and D, C is A, D and E, and D option is B and D only. So here students, we have the structure or the image of the cell, okay, and we have all the organelles. The question asked here is which of the following is not considered as a part of your endomembrane system, right? So we have organelles such as mitochondria, chloroplast and peroxisomes, right? So mitochondria, chloroplast and peroxisome, these three organelles are not considered as the part of your endomembrane system. Into the endomembrane system, we have organelles such as your endoplasmic reticulum, both smooth and rough. The nucleus is also a part of your endomembrane system. We have rhizosomes, lysosomes and other secretory vesicles as well as the Golgi apparatus. All of these organelles would be present inside the endomembrane layer and they are considered as a part of your endomembrane system, whereas mitochondria, chloroplast and peroxisomes are not into this part. So here the correct answer would be option A, because A, C and E, all of these three elements are not considered as a part of your endomembrane system. Here again students, we have to match list 1 with list 2. So into the list 1, we have different types of cells, that peptic cells, goblet cells, oxyntic cells and hepatic cells. And into the list 2, we are provided with the list of their functions, right? So we have mucus, bile juice, proenzyme pepsinogen and HCl, an intrinsic factor for absorption of vitamin B12. So let us find the proper combination here, and depending upon the combinations that we find, we have to choose one correct option from A, B, C and D. Okay. So here the first one, that is your peptic cell. As the name suggests, peptic cell which is present into your stomach lining, it is going to release a very important enzyme, proenzyme, that is pepsinogen. Because pepsinogen or this particular enzyme is going to remain inactive into your digestive system until HCl is released to activate that particular enzyme. Okay, so the role of peptic cell is to secrete the proenzyme. Here we have A along with 3, right? Now again let us try to eliminate any other option. So A3, yes, we can eliminate the 3 options over here and we can get the answer as 2, because no other option is going to show you the combination of A with 3. Then we have goblet cells. Goblet cells are usually present into our respiratory tract, right? So these kind of cells are going to secrete mucus that helps to trap dust and pathogens, right? Mucus is a very sticky substance, so it is able to trap all the dust and the pathogenic particles with the help of the goblet cells. Then we have oxyntic cells. Oxyntic cells are again important because it releases hydrochloric acid. Again, if you remember, hydrochloric acid is important for the activation of your pepsin enzyme, right? And it is also an intrinsic factor which helps for the absorption of vitamin B12 from your digestive system. Okay, and the last one that we have is hepatic cells. Remember students, ever you come across the term hepatic, that means something is related to your liver. Okay, so hepatic cells into the liver is going to secrete the bile juice again for your digestive system, which helps into the neutralization of the acid. So here the correct answer is your option B. So the next question here again is to match list 1 and 2. So in 1 we have Taenia, Paramecium, Periplantae and Ferentima, and in the list 2 we have some of the features over here. So we have nephridia, contractile vacuole, flame cells and uricose gland. So let us look at the options which are given to us, and depending upon the possible combinations you have to choose from A, B, C and D. So here the very first organism that we have is Taenia. So into this we have to determine what is the kind of a mechanism that they show to us, whether it's an excretory mechanism or an osmoregulatory mechanism for this particular cell, right? So for Taenia, it is the example that we have is flame cells which is going to perform the function. Now let us again try to eliminate our options. So yes, we can stick to option B because as we can see, none of other options is going to show you the combination here. Okay, so here for A, we can write it as 3. For option B, for the second organism, that is Paramecium. Paramecium shows a contractile vacuole. This contractile vacuole will help to maintain the osmoregulation of that particular cell. That means it is going to maintain water inside, that will help to take the keep a balance for the cell for the amount of water that they have and also the amount of salt or sugar that will have. Then we have Periplanta, which will have uricose gland again for the excretory mechanism, and we have Parenchyma, which is going to show the presence of nephridia, a similar mechanism for excretion from the cell. So out of the given option, students, the correct answer here would be option B. So here children, we are given below with 2 statements again. First is low temperature is going to preserve the enzyme in a temporary inactive state, whereas high temperature is going to destroy the enzymatic activity because proteins are denatured by heat. And the statement 2 is when the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of enzyme, it is known as competitive inhibitor. So depending upon the above statements or in the light of the above statements, we have to choose the correct options here. Option A that we are given is both 1 and 2 are false. Option B, 1 is true but the statement 2 is false. Option C, statement 1 is false but statement 2 is true. Option D, both statements 1 and 2 are true. So here let's talk about each of the statement. First, about the temperature. So freezing or storage, that means you are trying to reduce the temperature over here. This will arrest the enzyme's metabolic activity. Okay, the enzyme won't be able to function here. So as you can see into this graph, this determines the reaction velocity, or you can also call it as a rate of reaction, the speed at which the reaction occurs. Correct? So here this is going to determine the reaction velocity, and on this graph on the other axis we can see the temperature. So here the temperature is low and here the temperature is high. So at low temperature we can see that the enzyme activity is stopped here. The enzyme is not able to react or the enzyme is not able to function. Why? Because the temperature is going to arrest that enzyme activity. And as you can see, as the temperature is increasing, right? As the temperature increases, the rate of reaction or the reaction velocity is also increasing. So here we can see the increasing activity. And you would see the maximum reaction or the maximum activity at the optimum temperature here, right? So at optimum temperature, you can see that the enzyme will show its best reaction. Here, the rate of reaction would be the highest. But if the temperature goes beyond the optimum temperature, into this stage, you would see that the proteins are denatured because the peptide bond is sensitive to temperature. So if temperature increases, then it leads to denaturation. Thus, the activity will also reduce. So as you can see, as the temperature is increasing, the enzyme activity decreases. Okay. So here, this is our first statement. The second statement says, inhibitor is very similar to the substrate in competitive inhibition and it competes to bind the same active site. This type of inhibition can be overcome by adding more substrate to it. So see, here we have a substrate molecule which is binding with the enzyme. So they form together a enzyme substrate complex here. Once the enzyme substrate complex is formed, you would see that this particular enzyme would be able to produce a product out of it, because that's the role of your enzyme, to give a product out of it. But here, instead of the substrate, what we are doing is we are adding a competitive inhibitor. So ideally, the substrate goes and binds to the active site of the enzyme. This region is known as your active site. So the substrate molecule is going to go and bind to the active site of that enzyme. Competitive inhibitor is going to, block that active site over here. So instead of substrate, the competitive inhibitor is going to block that site and the reaction won't be happening. So here, inhibitor is a lot similar to the substrate in your competitive inhibition, because it is going to go and bind to the active site and it competes to bind to the same active site. So if you want to overcome this type of inhibition, you can add more substrate molecules. So if the concentration of substrate molecule increases, they will be going and binding to the enzyme. Okay. So here the correct answer is option D, that is both statement 1 and 2 are correct. So here students, we have to match the components from list 1 and list 2. Into list 1, we are given with the type of joints and the location or where exactly they are present. So we have cartilaginous joint, we have ball and socket joint, we have fibrous joint, and we have saddle joint. And we have between the flat skull bones, second is between adjacent vertebrae in vertebral column, between carpal and metacarpal of thumb, between humerus and pectoral girdle. We have to determine the possible combinations and then we have to determine the options from this one, A, B, C or D, depending upon what combination that we are getting. So here the first one, that is a cartilaginous joint, A. So cartilaginous joint is present in between your adjacent vertebra into your vertebral column, which is actually going to give the flexibility. Right now when you are moving the vertebral column, that movement is because of this cartilaginous joint which is present in between them. So here A will be paired with 2. Let us try and eliminate the option here. So A paired with 2. This time we are provided with into 2 options, A and C. But still we can eliminate our 2 options from the list. Okay, let's go and try again. The second that we have is ball and socket joint. So ball and socket joint is present between the pectoral girdle and the humerus, right, which is there into your shoulder. Right, here we have a ball and socket joint, this one. So the bone which is present in the upper arm known as the humerus, and here near the shoulder bone, that we call it as the pectoral girdle. So that is the ball and socket joint. So for B, we have option 4. So yes, it is present in both of the options. Still we cannot eliminate, but with option C we can. Then we have fibrous joint. So fibrous joints are actually thread-like structures, and that will be helping you to make the joint between flat skull bones. Our skull bone is not an one intact bone. There are small pieces of bone which are joined together with the help of fibrous joint here. So here the correct answer would be C, 1. And with this we can come to our final answer as option A, because C is paired with 1. Okay. Then we have saddle joint. It is present between the carpal and the metacarpal of your thumb. So into this thumb region, this kind of movement or locomotion that we are observing, that is because of the saddle joint, and it is present in the carpal and the metacarpal of your bone. So here D is paired with 3. This you can find into the option A. So here the correct answer is option A. So students, here we have to determine which of the following statements are correct. Option A, an excessive loss of body fluids from the body switches off osmoreceptors. Option B, ADH facilitates water reabsorption to prevent diuresis. Option C, ANF causes vasodilation. ADH causes increase in blood pressure. And ADH is responsible for decrease in GFR, that is glomerular filtrate rate. Here you have to choose the correct answer from the options which is given below. Depending upon which of the statements here are correct, we have to find out the correct statements. Option A is B, C and D only. Option B is A, B and E only. Option C is C, D and E only. And option D that we have is A and B only. So the first statement that we have, an excessive loss of body fluid, okay, that is from the body switches on the osmoreceptors. For example, into the hot conditions or into the hot climates, we tend to sweat a lot, right? So that is a way of our body regulating the temperature. And if the temperature is reduced, then the urination production is very high. Why? Because here your body is trying to increase the body heat. That's why it eliminates excess water. So here, when you are excessive loss of body fluid, when you are experiencing, be it into any way, that is why that the reason over here is your body has switched on the osmoregulators or osmoreceptors here. Into the statement B, ADH is going to facilitate reabsorption of water so that it can prevent diuresis over here. Okay, it will reabsorb the water. Then A and F can cause vasodilation. Vasodilation means where your blood vessels which are present near your skin, those are going to dilate, which will help to release excess amount of heat from your body. Then ADH causes increase in blood pressure because here the water is absorbed, water is reabsorbed, right? And as water will be reabsorbed from our urine or from our kidney, it will help you to increase the blood pressure here. Okay. Then you have ADH, which is responsible for increasing again into the glomerular filtrate rate. So here the correct answer is option A, because only a B, C and D statements are correct. Because here into E statement is responsible for decrease, which is incorrect because it helps us to increase the rate, right? And into the first one, it is switch off. Actually, it is switching on your osmoregulators or osmoreceptors. So the correct answer would be option A. So students, here again, which of the following statements are correct? Option A, basophils are most abundant cells of total WBCs. Basophils secrete histamine, serotonin and heparin. Basophils are involved in inflammatory responses. Option D, basophils have kidney-shaped nucleus. And E is basophils are agranulocytes. Then we have to choose the correct answer from the given options depending upon which of the following statement here is correct. Option A is C and E only. Option B is B and C only. Option C is A and B only. Option D is D and E only. So here ideally the basophil is not the highest amount of cells or most abundant cells which are present into WBCs. We have different types of WBCs, right? So here we have neutrophils and not basophils. Basophils are ratherly present in very small amount. Many times they are even absent into our blood system. Then basophils secrete histamine, serotonin and heparin. Yes, they help to secrete all of these molecules. Then they are involved into the inflammatory response. Whenever there is any kind of a injury in our body, let's say you had a muscle pull in our body, so it causes a swelling of that region. So at that time, inflammation is happening into that area. That is majorly because of the basophils which are involved into that inflammatory response. Okay. Then you have basophils which have multi-lobe nuclei. They don't have a kidney-shaped nuclei. Here there are multiple lobes which are present in the structure of basophils. So here we have the image, the neutrophil, the eosinophil and the basophil. Whereas neutrophils are granulocytes. We can see granules which are present in them. The granules which are present into the basophil, that is majorly because of the lot of enzymes which are present into the cytoplasm, which helps to create a response during the immune system, for the immune system here. So they are coming under the category of granulocytes and not agranulocytes. So out of the given options, the correct answer is B and C only, because here only these two options are correct. That your basophils secrete histamine, serotonin and heparin, and basophils are involved in inflammatory response. They do not have a kidney-shaped nucleus, they are multi-lobed, right? They are granulocytes and not agranulocytes, and the highest number over here is neutrophils and not basophils. So I hope this was helpful and you have understood it well. Thank you and happy learning.